Alright guys, so this is going to be video number 4. So this is where we left off. We left off asking ourselves this question. Did that recessive trait really disappear entirely? Or was it just maybe hidden underneath that dominant trait? And so of course Mendel decided to do another experiment. So what he allowed the hybrid tall offspring from that first generation to self-pollinate. So remember, he cross-pollinated on the first ones to create those tall hybrids, and this time he let them just do what they normally do and self-pollinate. So an F1 tall plant and an F1 tall plant created something that looked a little bit different. In that P generation, a true breeding tall plant and a true breeding short plant created all tall plants, and in this F2 generation, those tall plants created mostly tall plants, but also one short plant. And so they were 3/4 tall and 1/4 short. He found that that 3/4 of the offspring were tall and 1/4 were dwarf. The tall offspring had to have been carrying that dwarf trait, but it had been covered up by that by that dominant trait. So the wrinkled, the round seeds, the smooth pea pod, the tall plants were dominant over the recessive wrinkled, the recessive tight pod, the recessive short plant. So it was still there, it was just hidden. So that dwarf trait reappeared in the F2 generation. Just as a reminder, if the P generation is your grandparents, your F1 generation is your parents, and you would be that F2 generation. So why did the recessive allele disappear? Mendel realized that organisms have 2 alleles for every trait. These 2 alleles are inherited, one from each parent. If the offspring receives a dominant allele from one, then that dominant allele will appear in the offspring. No doubt about it. Recessives are only going to show up if the offspring receives a recessive allele from both parents. So in that tall short example that we did yesterday, anything that saw that tall in there anywhere is going to be a tall plant. But that short, the short would be short. So if a parent has 2 alleles for a trait, how can it pass on only 1 allele? This we have already covered. I'm going to go quickly through this because we covered it in depth yesterday, and I don't want to spend a whole lot of time on it so that you can use your time better for other things. Today we know that the answer lies in the type of cell division known as meiosis, or the formation of gametes. Gametes are, of course, sex cells, or an egg and a sperm. Looks like this. Here we're going to use capital and lowercase. I was really careful about yesterday writing out the entire word tall and writing out the entire word short. This is why we don't shorten things. A dominant letter, or a dominant allele, is represented by a capital letter, and a lowercase letter represents the recessive allele. So writing T and S would not have meant the same thing as writing capital T, lowercase t. So if we have big T, little t, then we replicate that DNA in the first interphase. So we have big T, little t, little t. We separate those homologous chromosomes in meiosis 1, put the big T's in one, the little t's in the other. Now we separate those sister chromatids in meiosis 2. Big T, little t, little t. During meiosis, DNA is replicated and then separated into 4 gametes. This way, the parent cell passes one allele of each gene into the offspring. This is the principle of segregation. Principle of segregation, really and truly, is just meiosis. We've already learned about this. I just call it something different. Mendel's principle of segregation says that every individual carries 2 alleles for each trait, These separate during meiosis, and which is the formation of the egg and sperm cells. So you're only passing on one, even though you start with two. Now, homozygous versus heterozygous. This is one of those places where students get confused. Make sure you're paying attention to this. Make sure you're focusing on this, because this is going to be important. You inherit two alleles for each trait. Those alleles can either be homozygous or heterozygous. If it's homozygous, what that means is that the two alleles are the same. Big T, little t. Big T, big T, or little t. Tall, tall, or short, short. If they are heterozygous, it means that they are different. So big T, little t, tall, short. And then genotype and phenotype is our last really big, really important, kind of hard to understand concept here. A genotype is the actual genetic makeup of the organism. Homozygous, heterozygous, those are your genotypes. Your phenotype is what you can point to. Your phenotype is the physical characteristic of the organism, what it actually looks like. So if you can point to it, it's a phenotype. If you have to use homozygous and heterozygous, if you have to use our science words to describe it, then it is a genotype. It is describing the actual genes. For example, in Mendel's pea plants, the tall allele was dominant over the short allele. So if we had the genotype, big T, or homozygous tall, then our phenotype was tall. If we had big T, that big T covered up the little t, so it was still tall, and then little t was dwarf. So that would be homozygous dominant, heterozygous, homozygous recessive. Same, homozygous, different, heterozygous. We're going to pick back up here on our next segment. Here in just a little bit, I'm going to get you guys to do one practice problem, and then you're going to be working some of the rest of these on your own.